Bismillahirrahmanirrahim. Welcome, Beta, in distant learning session, virtual lectures, and hope you all will be safe and healthy at your places, at your homes. And I pray to Allah to keep you safe, to keep you healthy from the pandemic, coronavirus, and all other kinds of infections. Good morning. Today topic is very important regarding the current ongoing scenarios, that is coronavirus, that mainly it needs prevention, and prevention in terms of uh, washing hands, keeping self cleaning and self protection, so sterilization. As our bodies are amazing structures and that defend us against infection under normal circumstances, as you can see in various pictures. In these pictures it's like dirty and everything is there, protection, the umbrella from the raining and also from extreme weathers. So we have a natural god given immunity that defend us from the infections. Whenever the body line of defense in terms of mucous membrane, mucosa, skin is breached due to any reason, any etiology, or during any operation, then this body's line of defense is breached, and then infection and so on, further damaging effects happened. So sterilization, disinfection, and asepsis becomes important. How we define sterilization? Sterilization, it is basically a process by which an article, surface, or medium is freed of all viable organisms, either in vegetative or spore state. This is called sterilization. And disinfection means destruction of all pathogenic microorganisms or organisms capable of giving rise to the infection. But drawback of disinfection is that spore forms may survive after disinfection. And if we talk about the methods of sterilization, we have two methods and two agents which can be used in sterilization, like physical agents and different chemical agents. In this slide, you can see various agents in sterilization, like physical and chemical, as we already talked about that. In the physical, the most important from the beginning of, you can say, this universe is the sunlight, then drying effects, dry heat from hot air oven, incineration, flaming, then moist heat from the boiling, pasteurization, sterilization, or autoclaving, then filtration. Filtration is radiation, ionizing and non-ionizing, and ultrasonic vibration. And in chemical agents, alcohol, aldehyde, dyes, halogens, different gases, they are used in sterilization. So we will discuss one by one. We will try to discuss all the agents or important agents. So first, physical agents, sunlight. This is a direct sunlight. This is a natural method of sterilization of water, especially in tanks, rivers, and lakes. This has active germicidal effect due to the combined effect of ultraviolet and heat rays. The next comes drying. 4 out of 5 bacterial weight is because of the water, due to the water. And what this causes drying, it causes protein denaturation and oxidative damage and toxic effects of elevated levels of the electrolytes. So the advantage, it can be easily employed, and disadvantage is poor penetration power. Physical agents, next is flaming. In this, flaming is basically the drying effect. Drying effects. The next is the first part of the drying is the flaming. This is the simplest method, and source of heat is Bunsen burner, and it is used in sterilization of the inoculation loops, wires, and tips of forceps or spatulas. Next is incineration. Incineration means bhatti, to burn the things. So incineration means burning the substance until it become ash in the presence of oxygen. This is an excellent method of destroying materials such as contaminated cloth, animal carcasses, and pathological materials, and uses in papers and cloths, especially in the cloths, infected cloth after the infected surgery or surgery done in hepatitis C or in HIV patients. Next comes in drying effects is hot air oven. This was discovered by Louis Pasteur in 1986, and it is basically a compartment with perforated trays and fans, and temperature is usually around 160 for 1 hour and preserve sharp edges of cutting instrument. These are the hot air ovens present in almost every theater. And articles to be sterilized: scissors, scalpels, glass syringes, swabs, liquid paraffin, dusting powder, fats, greases, and forceps. These all can be sterilized in hot air oven. Next, after the effects of drying effect is the moist heat. And mechanism of action is basically denaturation of the proteins and coagulation of the proteins, which lead to the killing of the organisms. It has three types of temperatures below 100 degrees Celsius and at 100 degrees Celsius and above 100 degrees Celsius. So we will discuss one by one all these. So pasteurization is basically temperature below 100 degrees Celsius. It has two processes: holders process and flash process, and it destroys mycobacterium, salmonella, and also brucella. And vaccines, non-sporing bacteria, heat inactivated in special vaccine bath at 60 degrees Celsius for 1 hour, and algae, media, serum, and other media which contain sugar and gelatin, they are sterilized in inspissor at 80 to 85 degrees Celsius for half an hour for 3 successive days. Next comes temperature at 100 degrees Celsius. Like boiling, vegetative bacteria they killed at 90 to 100 degrees Celsius, and time required is 10 to 30 minutes. It is not effective for sporing or spores of the bacteria. And when temperature is above 100 degrees Celsius, so steam under pressure, that is also known as autoclave. The principle is that water boils when pressure equals to the surrounding atmosphere, and saturated steam has more penetrative power. Two types of autoclaves are there. One is downward displacement and a pre-vacuum high temperature. How it is used, or time and pressure and temperature is maintained. If the temperature is 121 degrees Celsius, pressure should be 15 pounds and minute is 15 minutes. And if 126, then the same pressure but time is reduced 10 minutes. And if temperature is more increased up to 134 and pressure is 30, then only 3 minutes for the instruments to be sterilized. And articles used: surgical instruments, lab equipments, metallic syringes, and all culture media except those containing sugar and gelatin. Glass bead sterilizer. It is basically heat transfer device, and glass beads and salts to be sterilized. The articles sterilized are endodontic files and burrs. Temperature set is 220 degrees Celsius and time is just 10 seconds. It is useful for chairside sterilization and used in dental equipment, dental instruments. Sterilization controls. Control or how we check sterilization. There are different methods like autoclave tapes are there, and we can also check with the spores of bacillus stereo, thermophilus, or the agents used to avoid corrosive action of the steam, like ammonia we can use, and 2% sodium nitrite or ADT. Then gases can also be used in sterilization, that is radiation, Sterilization, it includes two types, non-ionizing and ionizing. In non-ionizing, ultraviolet rays are there, and ionizing radiation that includes X-rays, gamma rays, and cosmic rays. In all these slides, most of the data given or written in the slides is for MCQ's purpose, like how are the different types of ionizing radiation, or which radiation or which rays can be used in ionizing radiation, and you have to answer like ionizing and non-ionizing, X-rays, gamma rays, and cosmic rays. So first, in a non-ionizing, that includes ultraviolet rays. This bring down the number of microorganisms they are present in the air, and sterilization of the operation theater and biological safety cabinets usage are in UV rays. Disadvantage of UV rays is that it has a low penetrating power. And ionizing radiation like X-rays, gamma rays, and cosmic rays, cold sterilization is known, and they are very high penetration power, lethal to DNA and other cell constituents, so effective for heat labile items. The instruments which are used in sterilization is are syringes, swabs, catheters, needles, oils, grease, cannulas, metal foils, and culture plates. Next comes and the dental equipment which can be sterilized through the ionizing radiation, surgical instruments, sharp instruments, other materials, syringes. These all they like surgical instruments, autoclave, sharp instrument can be sterilized through the hot air oven. The other materials, including also the different instruments, autoclave and syringes through the irradiation, that is X-rays. Surgical handpiece sterilization through the gamma rays and sterilization, autoclave sterilization. Chemical agents in two forms, liquids and gases. In liquids, alcohol based and halogen based. In alcohol, ethyl alcohol, isopropyl alcohol, and halogens, iodine, chlorine. And in gases like ethylene oxide, formaldehyde, and glutaldehyde. Alcohol. Generally, using alcohols are as a disinfecting agents and antiseptic agents. We are using alcohol as antiseptic agents, and mechanism is basically denaturation of bacterial protein. And usually 70% alcohol is more effective than 90% alcohol. So low saturation, low percentage is used. The main uses are disinfection of clinical thermometer, or disinfection of the skin in venipuncture. And then the next comes in chemical sterilization is halogens. Normally using halogens as iodine and chlorine. Iodine in aqueous and alcoholic solution, this used widely as a skin disinfectant. Actively bactericidal with moderate against the spores, and also used for antiseptics on the wounds, different types of wounds. And chlorine, this is commonly used as a hypochlorite. So chlorine and hypochlorites are mainly they are mainly having bactericidal action, and they are used in different water supplies, swimming pools, and food and dairy products sterilization, cleaning. And if we talk about the gases, so ethylene oxide is used in sterilization. It is a highly penetrating gas and highly inflammable. Different articles which are used in sterilization, like heart and lung machine, respirators, different sutures, dental equipment, glass, metal, and paper surface. And in gases, the next comes formaldehyde gas. This is used in the fumigation of operation theater and other rooms. And after sealing the windows and other outlets, formaldehyde gas is generated by adding 150 grams of potassium nitrate to 280 ml of formalin for every 100 cu feet of room volume, cubic feet room volume. And then the room is closed, fumigation is done for 48 hours, and doors are open after 48 hours, and it leads to the sterilization of the operation theater. So this is all about sterilization, and different methods of sterilization and different articles which are used in the sterilization. So basically, this topic is mainly as for MCQs type and short SEQs. So thank you. If you have any questions, you can just please give me your feedback, and I will try to clear and answer your questions. So be safe at home. Thank you very much. Allah Hafiz.